Hi, my name is Angelika Amon. I'm a professor in the biology department at MIT. And I'm here today to tell you a little bit about me and about the science we do here at the Koch Institute at MIT. I was born in Austria, one of four kids, and I always loved biology. I always knew that I wanted to do something with biology. I wanted to first become a zoologist, a dinosaur scientist, and then in high school, I, for the first time, saw a very old movie from the 1950s that showed cells undergoing mitosis. You could see the chromosomes condensing into little sausages, and then they would sort of miraculously segregate. And I was fascinated by this movie. Is that this is so incredibly amazing. I need to understand how cells divide. And it was at that time that I decided to become a geneticist. I wanted to study the molecular mechanisms that underlie basic cell biological processes. At the time, I didn't even know that the father of genetics, Gregor Mendel, was in fact Austrian. So there was obviously Austria was the cradle of genetics. And even without knowing that, I already knew it was cool. And so I went to the University of Vienna and I studied biology. Initially, I studied zoology and botany. And then as I said, I became very fascinated by chromosome segregation. And for the second part of my studies, I did genetics and molecular biology. And then for my PhD, I joined Kim Naismith's lab, also at in Vienna at the Institute of Molecular Pathology. And it was in his lab that I first started studying the molecular mechanisms that govern cell division, especially mitosis. And it was in his lab that I got acquainted with the use and the power of the use of model organisms to study basic biological problems. So I came to MIT in 1994 as a postdoctoral fellow. Specifically, I went to the Whitehead Institute, which is one of the buildings that is part of the biology department. And originally, I was going to come for 2 years. And I was going to go back to Austria and sort of do research in Austria. But guess what happened? I fell in love with this place. I fell in love with MIT. And then when the opportunity arose to sort of stay here and take on a faculty position, I did not think once. I decided MIT is the best place to do science. It's the best place to do basic sciences. And if I get asked and have the opportunity to work here and do research here, I would be crazy not to take that opportunity. And so ever since, I took up my faculty position in 1999, and I've been here ever since. Since 1999, I've been a professor in the Department of Biology, and I still use budding yeast as a model system to study cell division. We basically are interested in the molecular mechanisms that make sure that cells divide their genetic material, their chromosomes, accurately. And we're also very interested in understanding what happens to cells in which this process goes astray, when all of a sudden an error occurs and the genetic material, the chromosomes, are not correctly segregated between the two dividing cells. I'm in the lab. I'm picking my yeast strains with a very high-tech tool, a sterile toothpick, and I move them from one plate to the other to give them more food to grow, right? And that's obviously a very easy way and convenient way to grow cells. It's much cheaper and much less finicky than growing mammalian cells. And so that's what we do. The way these cells divide, the basic mechanisms that govern cell proliferation in that yeast cells are the same as in mammalian cells, as in human cells. The processes are so conserved to the extent that you can take the human gene, you can stick it into the yeast cell that has a defect in its particular homologue, and the cells will work again. It will restore function. That's how similar the processes are that divide, that govern cell division across all eukaryotes, fungi, plants, and animals. And so I did my PhD in baker's yeast, the yeast that you guys all know that we use to brew beer, that we use to bake bread. And I have brought you a little plate here with the little yeast cells growing on it. See these little white little colonies? Those are yeast colonies that you use when you make pizza dough, for example, or when you brew beer. It turns out that many human diseases are caused by an incorrect chromosome number. For example, you probably all know Down syndrome is a condition where a human has an extra copy of chromosome 21. And we want to understand how having an extra copy of chromosome 21 affects the development of an individual, affects its abilities. You may not know, but it turns out that a disease that really plagues the Western world, cancer, a key feature, a key characteristic of that disease is having the wrong chromosome number. How is it that having the wrong chromosome number all of a sudden leads to cells proliferating in an uncontrolled manner, causing cancer? These are the kinds of questions that we are very excited about. And as I said, we are using the baker's yeast, Saccharomyces cerevisiae, as a model system to understand the basic processes of chromosome segregation and what happens to cells when these processes go wrong. What we also do is, once we've learned something interesting and important in budding yeast, we then move our findings to mammalian cells, to mouse cells or human cells, and ask the question, what have we learned in yeast? Is that also true in mammalian cells, and particularly in human cells? And in the vast majority of cases, that turns out to be true. Again, that's an illustration to the fact that the basic processes that govern cell proliferation are so highly conserved across all eukaryotes. I'm telling you guys, there's nothing better than doing science at MIT. Here, it's all about the science, and everybody is excited about the science. The students and postdocs work so hard day and night because they want to know. They want to find the answer to the particular question they're interested in. And that enthusiasm is just so infectious. And it's such a joy to come here every day and meet the incredibly talented graduate students, undergrads, and postdocs that work here and do science with them. I really have the best job in the world. It's quite remarkable.